Okay, hello everybody, and welcome to the HIRM seminar series for the middle of November. It's a pleasure for us to have today as our seminar speaker, Dr. Eric Miska. Eric is the Herchel Smith Professor of Molecular Genetics and a senior group leader at the Gurdon Institute at the University of Cambridge. He's now also the Deputy Director of the Gurdon Institute. He's also in the Department of Genetics at Cambridge University and an associate faculty member at the Wellcome Trust Sanger Institute. Dr. Miska received his bachelor's at Trinity College in Dublin and then his PhD at the University of Cambridge, making some of the seminal discoveries in epigenetics in the late 1990s. And then he was a postdoc at MIT with Bob Horvitz until 2005 when he started his own lab at the Gurdon Institute at Cambridge University. He is an EMBO Young Investigator awardee, a member of the EMBO, and a Hook Medal awardee from Cambridge. So it's fantastic to have Eric join us today, all the way from the UK. Thanks for that. The title of his talk today is going to be New Biology and Therapeutics from RNA Structure and Modifications. So welcome, Eric. It's good to have you here. Take it away.
Frank, thank you very much. Thank you all for joining in and participating in this new way of having meetings. I'd rather be there, but here we go. First challenge is, can I share my screen successfully with you?
Looks good.
Looks good? Okay, excellent. Right, so I'm going to tell you a little bit about the lab, and I'll be talking about two related stories. But as some of my hosts have reminded me already just before, I'm somewhat unfocused. So the whole lab is studying RNA, and we've been studying in particular RNA-mediated gene regulation, but that got us into RNA-based defense mechanisms and transposons. That got us thinking about viruses and so forth. And as you can see on the slide here, we also have diversified into a number of model organisms. Essentially, half of the lab is using C. elegans as a molecular genetics model, and then the rest of the lab is doing other things. And today I'll be talking about some of this kind of other stuff, really. But as a sort of baby introduction or kid sister introduction to RNA, of course, has been around for a while. And we were talking earlier, I actually run the Cambridge RNA Club, which is kind of sort of the continuation of a very old Cambridge RNA club where old men were wearing ties. But anyway, so RNA is made out of 4 different pieces. And we often obsess with the sequence. And of course, one of the major advances of molecular biology over the last decades has been the increasing speed by which we can sort of read RNA sequences. Super successful, super easy. But there is more to it. In particular, there is also RNA structure. And although I spent quite a lot of time working on very short or small RNAs, such as microRNAs and piRNAs and things like this, once RNAs gets longer, it gets more exciting because, of course, an RNA molecule doesn't like to be single-stranded. It tries to find something to interact with to form double helices, and then you get structure. And of course, the ribosome being extreme 1. And it is quite hard, in fact, to assess such structures inside cells, and I'll be talking a little bit about this. And I know it's a little bit early, but if we have a tree or a structure of RNA, we might want to decorate it as well. And I'm going to, towards the end of my talk, briefly talk about RNA modification. So how chemical modification of RNA, possibly even structured RNA, can change its function and why we should care about it. Okay, so the theme of the talk is to think about RNA structure and also about modification. What does this tell us about biology, and are there any therapeutic applications? And it's a bit of a boastful title, so I just want to, please don't get disappointed if I'm not going to present you with a new therapeutic in the end. And before I really get started, I just want to advertise something. You saw a number of weird model organisms, including honeybees early on. And if you want to read something a bit out of the ordinary to do with RNA structure in honeybees, I would like to advertise this paper we published last year. And I don't have time to talk more about it, but I think it's really a wonderful story involving RNA and immunity within a beehive. Instead, though, I want to talk about a new tool that we developed in the lab to study RNA structure inside living cells. I mean, we didn't start from scratch here. It's a further development of approaches that others have taken. But we think what we developed has really, major advantages over anything else out there. And I hope I can convince you that's the case. And before I go any further, I want to say that this is really the brainchild of one of the postdocs in the lab, Omer Ziv. And you published a first story on this in 2018, but I'll tell you mostly about what we're doing with this now. Okay, so it all starts with a molecule called psoralen. And I first got to know this at sort of the beginnings of the microRNA revolution, because there were a number of labs that tried to use psoralen to learn something about microRNA function. So psoralen is a double-stranded RNA cross-linker. And to be fair, to my knowledge, it wasn't very successful 20 years ago to help with identifying, for example, of microRNA targets, because to be fair, it's a lousy cross-linker of double-stranded RNA. And when I say these things, I'm not a chemist, I should admit, I'm just a geneticist. Anyway, it's a lousy cross-linker, and that didn't make it very successful. However, if you use such a tool like psoralen and combine it with something that's very efficient, such as high throughput sequencing, you can get some sort of cool results. And that's really what I want to talk about. Okay, and here I have to admit also that not Omer did come up with this technology in the lab, but also he's awesome at naming things. So, any decent kind of sequencing technology would have a short acronym followed by seq, but he insisted on naming this method comrades. And I mean, the best I can say about it is that, maybe because it's so horrible that maybe it's memorable. Anyway, so he developed this method called comrades, and it's using psoralen. And in fact, it's using a modified psoralen. And that's really one of the key tricks here, where we have click chemistry on the psoralen molecule that allows us to pull out of living cells cross-linked RNA using the psoralen as the hook, really. So the idea is that we want to understand what RNA structure and RNA-RNA interaction looks like in living cells. For example, here on the left, we might have this pale blue RNA, and it's an adventurous RNA. It has some hairpin loops in it, at least. And the idea is if we use psoralen, which can penetrate a cell membrane, so you can use this in cell culture, you can use it in tissues and even some organisms, although we haven't explored this to its fullest. Psoralen will get into these cells and will start making cross-links wherever you've got double-stranded pieces of RNA. However, RNA-RNA interactions don't have to be molecular only, but they can also be intermolecular. For example, we've got this little purple RNA here. It's kind of short. It's not very exciting on its own, maybe, but it does do base pairing with this pale blue RNA. So we get a piece of double-stranded RNA, and we can cross-link with psoralen too. In fact, what Omer is doing, he's titrating the concentration of psoralen to such a degree that we have approximately 1 psoralen cross-link for every kb or so of double-stranded RNA. So it is kind of, again, taking advantage maybe of this fact that psoralen is really quite lousy, if you will. We don't get many cross-links. And the idea being here that we are very gently probing the structure of the RNA without, essentially supergluing an RNA molecule in place, which might not reflect the structure in the living cell. And this concentration psoralen can penetrate cells, and these cells will remain living for quite some time. Okay, then the idea is we could we could study all the RNA after breaking these cells open, and this involves, in fact, some cross-linking with UV and reversal of the cross-linking. I'll skip over this and the details, but you can study, in fact, total RNA and just go for the cross-links, or you can focus on a particular RNA that you might be interested in. In this case, we use an aptamer approach. We make a library of RNA nucleotides complementary to the RNA of interest, and we can pull down very efficiently an RNA molecule of interest. And that's shown here in the middle. So because if the pale blue RNA is the RNA of interest, we have then aptamers. So we really tile the target RNA. It's a little bit expensive investment initially, but makes this process extremely efficient, and pull down this pale blue RNA down using aptamer, using biotin as a linker. After we've done this, we can fragment the RNA that has been pulled down, and fragments of RNA that contain a crosslink can then be subjected to a second pull down. And then we're going to do proximity ligation, if you will. So if you use 3C, 4C, 5C, high-C technology yourself, or maybe familiar with 3D genome structure studies that use this technology, the idea is you're ligating bits of DNA together that don't belong together, and you learn something about proximity. Here, the idea is very similar. You're doing this ligation of the fragmented RNA, and you're hoping that you make hybrid RNA molecules that contain different pieces of RNA from different parts of the same RNA molecule, or two different RNAs that are close together. And although I mentioned here this idea of proximity ligation, I would like to stress that sorrelin crosslinks only when you have a proper double-stranded RNA helix, so you wouldn't get accidental RNAs bumping into each other being discovered using this method. This method will only uncover RNA-RNA duplexes. Okay, so that sounds nice, and I'll try to advertise it, but does it actually work? And I'll try to convince you that it does. And here's kind of one kind of a bit of data, which is that if you do such an experiment, you could imagine even if you purify your RNA of interest, you're definitely going to get some ribosomal RNA with it, because there's quite a bit about. And in fact, just from sort of the leftover of the ribosomal R in any of the experiments, we get data for ribosomal RNA using comrades, and we can infer structure. And here is 18S ribosomal RNA from human cells, and there's a cartoon, a 2D cartoon of its structure. And whenever you see a colored nucleotide in here, that means that comrades has found direct evidence for base pairing. So you can see comrades covered quite nicely large parts of the 18S ribosomal RNA. And although some stem loops are blank, meaning there was no data to support this sufficiently, we don't have any illegal interactions, or we haven't found any interactions that are not known to exist from the crystal structure of the ribosomal RNA. So that's kind of good. It can find things that we know about, like the ribosomal RNA structure. But let's do some new biology. Let's try to discover some new things. And Omar and I thought that, and this is a couple of years ago, that really viral RNA is really good. It's a great model. You can just add it to your cells in culture, so you've got an easy negative control. RNA viruses tend to be quite structured and do interesting things. And there's some evidence, for example, for hepatitis C virus, that viral RNA can interact with cellular RNA to do interesting things. And partly for this reason, I was very excited to do this with hepatitis C virus. But then I was told by our health and safety department that I'm not allowed to. I work in developmental biology and cancer research institute, and they didn't think that I was set up to do hepatitis C virus. Anyway, so we got here this big list of viruses. We've got the DNA viruses and the RNA viruses. RNA viruses were more exciting to us because we could study the genome as well as the messenger RNAs from at the RNA level from the viruses. And we ended up narrowing it down to the flaviviruses. And I'll give you some of the reasons in a minute, or hopefully it will become clear. If you look a little bit to the right, you can see here corona as well, and I'll get to that later on in the talk. Okay, so flaviviruses we're allowed to work with, or at least one particular flavivirus, which is actually the least harmful of these guys, and this is the Zika virus. So here's some cartoons of 4 related flaviviruses. that have an impact on human health: Zika, dengue, West Nile, and yellow fever. And not only are there flaviviruses, I hope you can see in this cartoon at least that they also have some structural similarities. In fact, people have been studying the structure of some of the RNA genome of these viruses for decades. This is usually done in vitro and usually using parts of the virus. But at least we thought that we had some information that we could compare our new method with. If you look at this, though, you might think, is there anything new that these guys can discover? And the secret is in this little yellow bracket that is shown here, because what is really going on is this, that the structure that is known is only for the very 5' end and the very 3' end of these viruses. There is no structure known for the whole coding sequence of the virus. Furthermore, the structure that we do have is only derived in vitro, not inside human cells. And also what was not known for these viruses, if the viral RNA can interact with cellular RNA. And we thought, we now had a method to test this. Okay. So we used comrades by infecting human placental cells, or a cell line, human placental cell line, with Zika virus, and then carried out this comrades method. And you can try to visualize the data in a certain way. Here's a very simple way where you do a diagonal plot. Flaviviruses, and that's one of the reasons we chose them, are kind of simple, relatively short genome. So here you see about 11 kb of the Zika virus genome in the X and also in the Y. It's a diagonal plot, so any spots off the diagonal suggest some sort of interaction of pieces of RNA that are apart from each other, right? And in the zoom in, you can see it kind of blobby. And you look at this and you look a little bit more and you say, well, there's something there, but does it mean anything, right? And to convince ourselves that it does, we try to look at replicates. So essentially, Omer took human placental cells, infected with Zika, and then went through this whole comrades method, did the sequencing and did this plot. And he did this a number of times. And this is what the result looks like. And it looks almost too convincing, but I tell you, these are real 3 independent replicates. So replicate 1, 2, and 3, independent cultures, the whole procedure done. And these are kind of the spots that we find. And there are also 2 control experiments. Omar dropped one, to be perfectly honest, where we don't see these spots. So we don't do the cross-linking here in these control experiments. Okay, so what we have here is, I would say, at least a reproducible method. Of course, that could mean that it's reproducibly wrong. But the only defense I have for this is that I showed you earlier the ribosomal RNA structure. At least for the ribosomal RNA, it got a lot of the things right, and in fact, nothing wrong. So we are hoping that for Zika here, the same is true, that these spots represent real interactions. But let's have a look at what kind of biology we were able to uncover. First of all, again, and I apologize if any of you are real virologists. I'm not, I'm just an RNA biologist, so I'm trying to be. Anyways, these viruses have been proposed to circularize inside cells. And the idea is that being in a circle or being open for the viral genome is important in switching between 2 major tasks that the virus has to accomplish inside cells. First of all, the RNA is, of course, the template for translation, and you need to make viral proteins, right? And the other thing you have to do is you have to copy the viral genome using an enzyme encoded by these viruses, an RNA-dependent RNA polymerase. And the idea is that the replicating viral genome forms this beautiful circle as indicated here, and that the translating viral genome is in a linear extended form. And in fact, there are 2 particular regions, the 5' circularization sequence and the 3' circularization sequence. And these are the regions of the genomic RNA that start this zipping up to get you to the circular form here. Okay, this has been inferred indirectly by mutational analysis, a number of other experiments, but nobody has kind of seen this in living cells. So we thought, okay, we could at least test if this is indeed the case. Do we find any evidence using our method for base pairing in this 5' circularization sequence and the 3' circularization sequence? And the short answer is yes, we do. Here's actually the raw data plotted as diagonal plot. Doesn't look very exciting. There are just these blobs here, but these blobs in fact correspond to the 5' and 3' circularization sequence interacting with each other. And here, down here in these linear plots, it's shown slightly differently. So the question is, if you take the 3' circularization sequence and ask, do we get any hybrids along the 5' end of the virus, then we see one very strong peak in one locale on the RNA genome, and that's where the 5' circularization sequence is. And if we take the 5' sequence and look where does it interact with other parts of the viral RNA genome, we see one peak at the 3' circularization sequence here. Down here are the controls. So we see, in fact, RNA-RNA base pairing with these two sequences inside cells. That seemed good. Also, I mentioned before, and here shown again as a cartoon, previous work in vitro showed that there's a bunch of hairpin loops and pseudoknots in the 5' and 3' UTR of the virus. And we were able to identify or get data for all of the pseudoknots that have been described in Zika virus. And here are some examples. Again, the color indicates actual base pairs detected using this comrades method. So that seemed good. What about interaction with cellular RNA? And so we were very excited about this, and we had some amazing hypotheses how important this might be. But here's what we got. To start off with, we didn't find any evidence for longer RNAs, long encoding RNAs or messenger RNAs to be base pairing with the viral genome inside human cells, but we found evidence for some microRNAs, for some tRNAs, and also for small nuclear RNAs. Curious. And to be honest, it was a bit disappointing. First of all, microRNAs. Some of you might know that hepatitis C virus, Peter Sarnoff's lab had shown a while ago that a particular microRNA base pairs with the hepatitis C virus, and it's important for the function of this virus inside liver cells. So finding a microRNA was maybe satisfying, but not absolutely shocking. There's precedent. tRNAs, we initially thought that's a bit suspicious. You know, is this just something where the ribosome got stuck and we're cross-linking tRNAs? It turns out slightly more interesting because in fact, these are tRNA fragments and very specific tRNA fragments. Although unfortunately, we have still not worked out the functional consequence of this interaction. And also very specific positions where you get small nuclear RNAs. That sounds a bit funny because small nuclear RNA spend most of their time in the nucleus. The viral genome is in fact in the cytoplasm. But nevertheless, we think there might be something really interesting going on here with the viral genome marking itself by using these small RNAs. But again, unfortunately, we don't have any functional data to discuss this further. We did some functional follow-up on this particular microRNA miR21. I show you only this microRNA. We found a few other microRNA interactions, but miR21, I think, was about 100-fold stronger. There was 100-fold more evidence for cross-linking between miR21 and the Zika virus genome than compared to any other small RNA, in fact. Okay, where does it bind? Well, isn't that a bit funny? So the human miR21 seemed to be cross-linked by sorolin to one particular position at the 5' end of the virus. And again, here in red, you see all the direct evidence we have for this cross-linking. But I spent quite a few years of my life thinking about microRNAs and microRNA targeting. And probably Frank could definitely agree with this. This doesn't look very good. Okay, so here is the 5' end of the virus. And here's the 5' end of this microRNA, and there's some base pairing there, but ooh, this doesn't look good. This doesn't look like a classic interaction between a microRNA and a microRNA target. So we're kind of skeptical that, maybe this is kind of an artifact we're following up here. Therefore, we started a collaboration with Ian McGray, who was the first to crystallize human argonaut with an RNA in it. And we asked him to test this interaction in vitro. And in fact, what he did is he made human miR21, loaded it into human argonaut, and then tested in vitro interaction between miR21 in an Argonaut and this 5' end of Zika. And indeed, he found a specific interaction between Zika and miR21 in this position, and it was abolished by mutation of the microRNA or the virus. Okay, so this seems to exist. It is kind of a curious interaction. If you can, I don't know, on my screen I can see it. I don't know if it works via Zoom. But you can also look it up in the paper. In fact, there's more base pairing here that could happen between the miR21 microRNA if it would be able to melt this stem loop here in the Zika virus. But interestingly enough, we didn't find any evidence from the comrades cross-linking that this is in fact happening. We find only very strong evidence for this interaction here. Anyway, this seemed interesting. First of all, a new microRNA binds a new virus. We didn't know about it. And of course, you might have spotted already annotation here, miR21, and I just thought this is just too good to be true. It interacts exactly with this 5' circularization sequence that I just mentioned, right? So this is exactly this part of the virus that needs to interact with the 3' end of the virus to form the viral circle. So obviously these two events are mutually exclusive. And therefore then we hypothesized that miR21 microRNA might be involved in titrating this switch from the translating virus to the replicating virus. At least to my knowledge, we don't know how the virus is doing this. Wouldn't it be a reasonable hypothesis to assume that miR21, which is present in many human cells, certainly in all the cells that are part of the tropism of Zika virus in human, use miR21 as a yardstick to measure how much of the virus is present inside the cell, which would affect the relative interaction, either circularization or binding to miR21. Okay, but that would mean that messing with miR21 should affect viral replication. And we have some evidence to support this, and I'll briefly go through this here. And I should say it's not a gangbuster effect. You know, doing something to miR21 doesn't give you a 100-fold difference in the infection rate of Zika, at least in tissue culture. But there is a, there is a significant and reproducible effect. And here it is. So first we took our human placental cells. These are the wild type cells. And we simply now measure viral infection by qRT-PCR of viral genome. And we made two independent CRISPR knockouts of the single miR21 gene in the human genome. And if we do this, viral infection go down by twofold. Okay. In a parallel and sort of, somewhat weaker experiment, but we thought we should give it a go. We use here a conventional LNA inhibitor, so an antisense modified RNA oligo to sort of inactivate miR21. And here's a control inhibitor, and here's the miR21 inhibitor. More or less looks the same. So you get a twofold downregulation. Okay, there's an effect. It's reproducible in a number of different cell types, in fact, but this could be entirely indirect, right? So can we provide any evidence that is because miR21 might be interacting here? And we tried to address this, and what we did use is a viral replicon that had been mutated. Because if we think about it, if really the main role of the 5' circularization sequence is to bind to the 3' circularization sequence, that means if you mutate one of the two, then the virus won't be able to replicate, and that's in fact the case. But if you mutate one and then do compensatory mutation in the other sequence, you should be able to fix this. And this is in fact true. And you can make a replicon that can still replicate in human cells this way. But there is then a difference between the wild type and the mutant replicon in that one should be sensitive to miR21 inhibition and the other one shouldn't. And that's exactly what we see. The wild type replicon that can interact with miR21, we see again this twofold reduction of viral replication if we add the inhibitor. Because this replicon has a luciferase gene in it, we can just use a luciferase readout. And if you use this mutant replicon where miR21 can no longer bind, adding the miR21 inhibitor has no significant effect on viral replication. So we propose that miR21 acts to aid Zika virus replication inside those cells by directly binding to the 5' circularization sequence. So you can imagine then there's this sort of circle of life of the virus, and it uses miR-21 to titrate itself. Now, this is just a little bit of crazy talk on the side, but what's miR-21's normal role, really? Well, it's a bit of an enigmatic microRNA. I think Dave Patel published a while ago, and maybe others too, miR-21 knockout mice even, and they're doing okay. miR-21 has been suggested to be an oncomere. Its target space is kind of poorly characterized, but maybe some of you know more about this than I do. But I just noticed here, because actually there's been this curious link between between Zika virus infection and certain types of brain cancer. Anyway, if you heard about it, think about it a little bit more. If you haven't heard about it, forget it altogether. Okay. So I started off with talking about trying to understand viral RNA and possibly viral RNA structure or behavior in living cells. And, all I'm showing you is weird diagonal plots and then claiming certain structures exist, but, seeing is believing. And so we thought it would be really great to use an orthogonal approach to at least look at some of the things we found and see if we could find this to be true as well. So what we were able to do was to collaborate with the lab of Daniel Zenclusen in Montreal, and we sent him slides of Zika virus infected human placental cells, and he made highly sensitive probes for the 5' UTR and the 3' UTR of Zika virus, and we looked inside cells what's going on. In fact, what is going on is that you might be able to see the two kinds of states of the virus. If you look here, it looks like the red and the green are very close to each other. You have to take, a bit of, of course, this slide doesn't prove it, but we have, looked at this quite a bit. So you see the red and the green come together, almost if we're looking at a circle here. And here you see maybe the red and green further apart. So maybe one is translating and the other one is replicating. You also see this kind of thing. What's going on is a bit suspicious. And I've been asked, could there be viral concatemers? And I'm not aware of any evidence for this. And to distinguish between the two, I think we would need some mutation analysis, maybe use 2 strains of the virus where we could distinguish this, but we haven't done it. But it's an interesting idea, but as far as I know, no evidence that there is possible concatemerization. Right. Okay, flaviviruses have one more trick up their sleeves though. And again, it was one of the reasons why we picked them. Okay, flaviviruses have 2 hosts, an insect and a mammal, unfortunately often us. And that means that the virus has to live in 2 very different environments. If you're an RNA molecule to start off with, the mosquito is a little bit lower temperature than the mammal. And the cellular environment is different. I've just talked about the miR-21 microRNA, for example. There's no such thing in insects. So we wondered, and that was kind of the idea from the beginning, does the structure of the virus change in different cellular environments? And does it use different structural tricks, if you will, in the mosquito environment and the human environment? Okay, so to do this, we got Edie's cell line, so a mosquito cell line, and it has somewhat impaired RNAi pathway, so you can pump these guys full of Zika. And what you see here reminds me a little bit of microarray technology from the last millennium. The idea is we're looking at 2 comrades experiments, one in mosquito cells and one in human cells. And whenever you see yellow, you have the same structure in both cell types. And if you see a red dot and a green dot, you've got different structures. And you might look, it looks pretty yellow to me. But in fact, there are some green spots and there are some very red spots. And there are a number of specific differences of the structure of the virus in both systems. And again, unfortunately, I have to say that we haven't done any functional follow-up yet. It's very exciting. We have identified a mosquito lab that we can work with, and we're working on it, but no data so far. The only thing I would say, though, is that in the mosquito cells, we find some long non-coding RNAs interacting with the mosquito genome, which we're quite excited about. I mentioned earlier that we have no such evidence within the human cells, but in the mosquito cell, we've got a long non-coding RNA directly interacting with the viral genome as identified by comrades. And we're trying to figure out why the virus would be doing this, or possibly even the mosquito. Okay, that's it. But maybe it was obvious where this was going, right? So we had developed this technology in the lab. There are a number of projects now going on. We're looking at long non-coding RNAs. We're looking at the structure of the ribosome. You know, I thought we knew this, but actually it's quite interesting to figure out maybe how the ribosome biogenesis, how the folding works. So we're working on this. Comrades is brilliant for this. Some other RNAs that we're interested in, and maybe sort of, you know, maybe to sort of maybe think about where I want to end. There's quite a bit of interest in RNA structure as a new target space for small molecule therapeutics. And if that's something you're interested in, maybe you should find some new RNA structures inside cells. And I think this method is exquisitely good at doing this. Okay, but of course, then, 2020 happened. We were quickly shut down. And the only way we were able to get back into the lab is coming up with some excuse. Now, I'm not trying to belittle it, but of course we were super excited to apply this technology to another RNA virus that was rather topical, which is SARS-CoV-2, and it's now available online. We did a few months ago solve essentially the structure of SARS-CoV-2 in monkey and human cells. Okay, now coronavirus are not like flaviviruses. In fact, they're the most complex RNA. They have the most complex RNA genomes, and there's not only one RNA molecule, and that's the genome. That's also the mRNA. But in fact, through some processing, you end up with a genomic RNA that is, of course, required for replication, and then we end up with messenger RNAs that are all extracted from this original genomic piece of RNA. So not only would we want to get the structure of one RNA, but of all the RNAs. Okay, so we applied this commerce. I'm just skipping over some details, and as I said, the paper is on the archive and also on Molecular Cell. They have got a SARS-CoV-2 special online I don't know, resource, I guess. But we applied essentially the same technology, being a little bit more careful with the virus than we were for Zika. And first thing to say is that the structure of the gRNA is quite different from the structure of the subgenomic messenger RNAs. And I'm not going to show you any detail here, but these kind of peaks refer to hybrid RNA hybrids that we're detecting by comrades, and they look quite different. Furthermore, we're detecting a lot of very long-range RNA interactions. Now, for Zika virus, I talked about the circularization event, right? So seems like that seems reasonable. It's a good example of a long-range interaction. And comrades is great at figuring these out. But actually, some of the other tools that are used to sort of infer structure inside cells or in vitro are really not good at getting these long-range interactions. But for SARS-CoV-2, again, like with Zika, we find some very exquisite long-range interactions. And as I said before, there are huge differences between the subgenomic messenger RNAs and the genomic RNAs. And we cataloged them all. And again, Omas did strike again. So he invented a comrades method, and he named all these RNA loops arches. And we had a huge debate in the lab if this is an arch or not. Whatever, now it's an arch. We're calling it an arch. And here are some of these examples, right? Where you really see, look at this guy here, there's a 30 kilobase loop where we have got very strong, so these are only, very highly selected examples where we've got huge amounts of evidence from the method. And in some cases, we got also covariation data from the coronavirus genus, if you will. to support this, we've got very strong data that these structures exist. And of course, I would say that this not necessarily mean that all of these things are functional. But again, I'm not a virologist, but a virus has very little RNA sequence. It can evolve quickly. If things like this hang around within a virus, they're gonna have some functional role. At least that's what I would suggest. The most curious thing we found was when we looked at the so-called frame shifting element. And I didn't talk much about SARS-CoV-2 in this talk, and you might already know much more about it than I do, or maybe don't really mind. But here's kind of a cartoon of the genomic RNA, and you've got this ORF1A and ORF1B. And it's not like that I'm very bad at PowerPoint, but this step here is suggestive and reflects a frame shift that is happening. So these two open reading frames are in fact there to make polyproteins. So a number of proteins are made from this polypeptide, but you can either make A or you can make A plus B, and to do this you need to, or the ribosome needs to perform a frame shift, a -1 frame shift here. How does it do it? And it was known before that there is this pseudoknot here, and that's what's called the frame shifting element. The idea is that this pseudoknot is a pretty sort of neat little thing that is involved here. But when we looked at this part of the viral genome using, using the comrades method, we saw this huge cross that suggested something really a big structure. And in fact, there's a giant arch surrounding this frame shifting element. And again, it's kind of 2D is a very poor cartoon, but look at this. So it's essentially like a 15. So exactly in this region here, we've got essentially a 1500 nucleotide super loop, which we try to draw out here using. I mean, there was no structure of this before, but this is purely a predicted structure on the comrades method. This is what this looks like. I mean, this must be important for the regulation of the virus, and we'd like to find out more. But again, I don't have functional follow-up data as of yet. The only thing I would say is, again, if you look in other coronaviruses, look at sequences, you can spot. We look for covariation supporting the structure, if you will, and we have some evidence here and here. But even more exciting, we took advantage of data published recently by Andrew Firth's group, who looked at conservation among the coronavirus family, and the regions that are involved in this big arch here, where the frame shift element is. This is kind of this big loop here, is under very strong purifying selection. These sequences are the most highly conserved sequences within the virus. Viruses are kind of complicated, right? Because of course you need conservation also for the protein sequence, but this is way beyond. So we think there's extremely strong indicative evidence that this structure is very important for the virus, and we'd like to know more about it. In particular, I would really like to see that structure in 3D rather than this poor little diagram we have in 2D. Right. In addition, I already mentioned family of coronaviruses, but we have evidence also for this in MERS. So you can see this cross here. We have much less data for MERS. Essentially, it was a pilot study, but we'll have more data soon. We think this is a highly conserved structure across coronaviruses. Okay. This is when I go in the morning to my office. That's kind of what I see, although it's usually not sunny, of course, so you have to be lucky. And just to zoom out for a second, Zoom is terrible, no? But anyways, I just want to switch tack a little bit and spend a few minutes for pure advertisement. And this has to do with RNA modification. And I'm sure it hasn't escaped your attention that there's a lot of excitement on chemical modification of RNA, which we knew for a long time, but we also don't know much about the function of many of these modifications. And of course, my lab has been excited about this also. Personally, we worked a lot of modification of M5C and derivatives, for example, M5CM, something we are very interested in, but I won't have time to talk about this. But probably the best known modification, and some of my kind hosts are working on this, of course, is the modification of M6A, where there is tremendous evidence that emerged over the last 10 years of its role, in particular in messenger RNA metabolism. And what is kind of neat and brings me back to my early life when I was a PhD student working in epigenetics, that it seems to have some sort of similarities to histone modification, where you've got enzymes that put the modification on, you've got enzymes that take the modification off, and you've got reader proteins that sort of can bind to the modified but not the unmodified RNA. Anyway, a lot of excitement, and I'm just introducing this because I got very excited about this tour a while ago, and together with my colleague and mentor Tony Kouzaridis, we co-founded a company now in 2015 that tries to use this sort of family of enzymes that modifies RNA as therapeutic targets. And I don't want to bore you with this. I just want to share one bit information in case you're interested. And as I said, I'm a founder and director on the board of this company. So please treat this as pure advertisement. Here are some of the guys involved. I just want to show you this thing in case you care about RNA modifications. And Storm is still interested and open to collaborations on this. The idea is that the company has pushed RNA mass spec to the extreme. And it's not perfect, but it really has been a tremendous step advance, really. The idea is to use RNA mass spec, similar how you would treat proteomics, right? If you do proteomics, you don't, essentially cut all your proteins into amino acids and ask your mass spec machine to tell you how many lysine residues is in the pot, but you're interested in the sequence of the protein, so you just digest the protein into peptides, get the peptide information, and then can work out what kind of proteins you're dealing with. The challenge is the same. Almost all mass spec done on RNA is actually in fact not done on RNA, but it's done on nucleosides after complete digestion. And then you can count how many M6A, M5C nucleosides do I have in my RNA bag, which is not as informative as this stuff over here, where you use specific enzymes to digest the RNA into oligonucleotides, then put them into the mass spec and get sequence information, plus, of course, possible modification. And as far as I know, Storm is the first team that got this working with cellular RNA. It doesn't work for a very rare RNA. It's not perfect, but it seems very exciting. It has led to quite a number of new exciting starting points for research in the lab. And if you're interested, please let me know afterwards or get in touch with Storm. Okay, I'll skip over some other stuff since it's already late. And over Zoom, I find I'm talking much slower. Maybe I should stand up. Anyway, we did this work with some amazing collaborations. Almost all the stuff that I talked about was led by Omar Zif, a postdoc in the lab, and he worked with Sveta, a master's, now a PhD student in the lab, and John Price, who's the computational guy. We collaborated with some amazing groups, and among all of these wonderful people, I mentioned some already, but I want to also highlight in particular Friedemann Weber, who's a virologist that I met a very long time ago. In fact, just after the first SARS outbreak, and I called him up during this outbreak saying, okay, are you still working on coronaviruses and can we do something together? And it was because of him that we got started with that we got started really in the lab doing the SARS-CoV-2 project, which really got us through the lockdown. And I'm going to show you this video again. And just to say that, you know, science is scary. You have to be brave, especially in my lab. Here are the guys. And as I like to say, there's always more space in the boat. So if anyone's interested in coming to a crazy place in England, please get in touch. Thank you very much. I think I'll stop sharing my screen now.
Thank you very much, Eric. That was really great. Really appreciate it. So we have some time for questions. If people would like to ask their questions in person, please unmute yourself and ask your question. Otherwise, feel free to type your question into the chat and then I will ask for you. So The floor is open for questions.
Don't be shy, please. I would love to answer at least one question.
Well, Eric, maybe while people are thinking, I'm going to ask a question. So, you mentioned the conservation between the coronaviruses, but what about the flaviviruses? So, I mean, is miR-21 looking like it could be binding the 5' end of, dengue and yellow fever and other viruses of that class?
God, my God. I mean, it's like, my God, I think I need to replenish my memory banks. I don't think so, but I wish Omar was here now to shout if I was saying the wrong thing, but I don't think so. No. Right. A lot of the 5' and 5' and 3' UTR pseudoknots are conserved, but I don't think miR-21 is conserved. And the tropism of some of these viruses are quite different as well. So no. Right. So miR-21, I don't think it's going to be a cure against dengue. And if I said this now wrong, Omar's going to kill me. But I think I remember this correctly, I think.
All right, thanks. Any other questions?
This is Jean Schaefer. I have a question. So you showed us beautiful evidence of the power of comrades to detect these interactions. And in your first example, you showed us the 18S ribosomal RNA and how well the approach worked to predict the known structure. In your studies of the 18S RNA, did you find interactions for snow RNAs with the ribosomal RNAs? And can you comment on how long-lived the interactions have to be for this technique to actually reveal them?
Yeah, so for snow RNAs, yes. And we're quite excited. So we're just down the road from Venki Ramakrishnan's lab, and we're trying to see if we can look at some of the funny things that we found, maybe. But we haven't done this yet either. I mean, we really delayed with a lot of things. With regards to how, essentially how long an RNA duplex has to be around for, I really don't know. It's very, very hard to say. And of course, I didn't emphasize this specifically, but like any of such techniques, there are heavy biases in it, and it's very, very hard. I mean, it's, we often say, wow, we've got a lot of evidence for this interaction or this stem loop, but actually, does this mean that this stem loop is more stable or hangs around for longer? It's actually really, really hard to say. What I didn't talk about with regards to Zika is that we also found, in addition to the open and closed form, we found a number of alternative structures that are incompatible with each other inside sort of this transfection experiment. And the way we would love to interpret is that we're actually seeing more dynamics, right? That the virus can have these different alternative structures, and we see them, but it could, and what is nice is that some of the most frequent structures we do, think we see, are also thermodynamically favorable, but we could just say it's kind of messy as well. I mean, I think we really need follow-up using other techniques, in particular for RNAs that are less studied. Yeah, I don't know. I better stop here. I don't know. But if you are interested, please try it. I mean, if you want to try it out for whatever problem you're working on, I think could be quite good. It works. It's very simple, very robust, and
Eric, I have a question. Very nice talk. Thank you. So my question is about obviously it's the more relevant structure is the one in cells, as you showed, but like, how much is the structure impacted by cellular proteins or viral proteins? Have you done a comparison of the RNA, like in vitro, just isolated RNA versus the structures you're showing, and how similar are they?
Yeah, we haven't done much of this, to be fair. Some essentially the 5' and 3' UTR, they are very sort of baked in pseudoknots, and they have been found in vitro as well. I think, it would be quite hard to make this huge RNA in vitro, and I don't know, I would be surprised, but we haven't done it. I would be surprised if it would be just the same, but we haven't done it, to be honest. With regards to RNA binding proteins, what is missing now, and that seems very exciting to look into, is to combine this method also with RNA binding proteins. And of course, if a protein binds an RNA, then it might not be available for RNA-RNA interaction. So you know, this might be quite a nice data set to overlap. The same with the ribosome, the RiboSeq kind of data. I think that would be also exciting. And we're trying to do this right now because luckily other labs have generated a lot of these data sets. But yeah, so yeah. But exactly, that's exactly right. I'm hoping that, cellular environment, not only RNAs, but RNA binding proteins would enforce or force certain structures and that we could detect this.
I noticed there's a couple of questions in the chat.
Yeah, I'm just waiting for people in the audience to ask their questions first. So I think, Anna, you had a question? Go ahead.
Yeah, sure. Hi, Eric. Thanks.
Hello.
Hi, good to see you. So a couple questions. I guess first is whether you try to develop any imaging-based, or if you're aware of any attempts to develop imaging-based technique to see, to detect intermolecular interactions within the RNA, maybe based on fish hybridization or something like this. Do you think it's feasible? It would be interesting to try and validate some of your interactions using something totally different.
Yeah, I mean, absolutely, yeah. Yeah, and we would like to do a little bit of this. As I showed, I mean, we started working with Daniel's inclusion a bit, but what I would really love to do is get my hands on sort of an organoid system and look at SARS-CoV-2 infection and see maybe that at different stages of infection, we might see differences in structures. So for this, what we need to build, based on the comrades structural models, we would build particular probes to probe the existence of certain structures, maybe early in infection, later in infection, and certain cell types versus others. That's kind of an idea, but it's hard to, I don't know what you guys are thinking, but it's very hard to, crystallize or come up with crisp ideas over Zoom meetings and then to start new things. So it's all still in a preliminary stage. It's so hard.
Judy, you have a question?
Yeah, I have a question. Hi, Eric.
Hello.
So people working on coronavirus use anti-double-stranded RNA as a way of distinguishing actively replicating virus from, passively acquired virus. And I was wondering, from your structure, do you have any insight into what these double-stranded RNA regions are? I don't know how big they have to be to bind to the JP antibody, but it's just curious.
Yeah, so I would say I'll wait. So what Omar's doing right now is that so I've only so we purified specifically the RNA, genomic RNA and its sort of descendants. We also will now probe the opposite strand, and we haven't done this. So essentially, I think we are a bit disadvantaged so far because we're pulling down single-stranded RNA. So when the virus is actually replicating, it would be very hard for us to pull it down using an aptamer approach, right? Because it would be double-stranded. And I think the way to control for this is to pull down the other strand.
Well, you could use this anti-double-stranded antibody to pull down selectively.
Ooh, yes, that's we could look at this, yeah. But at the moment, our idea was simply to get the negative strand, pull it down, and ask what structures do we get from this, and then infer. The double-stranded RNA antibody sounds very exciting, and in fact, we do have a wonderful collaboration with a lab on this, but yeah, we don't have any data on it. But yeah, it's a very interesting idea. What I did find fascinating is that if we look at the structure we see, it maps very well with issues of being able to sequence. When you sequence the virus and say, ooh, this region is very hard to sequence, and it matches quite well. You could argue, well, everybody who's trying to sequence already knows the structure because they run into these bumps. But yeah.
Thank you.
Sean?
Yeah, so Eric, that was really interesting, and I guess very exciting, and lots of opportunities with things to do with it. But I was just wondering, in the context of, for example, oncolytic viruses and potential of kind of looking at how oncolytic viruses might kind of be interacting with genomes in terms of cancer cells, normal cells. and maybe also in the context of engineering oncolytic viruses to be maybe, enhance those kind of targets or things in that way? Have you thought about anything in that way?
Yeah, sounds super great. All I can say is I only got one postdoc working on this, but he's kind of fully occupied. But I think he'll be very excited for to collaborate with others. Yeah, okay. So one thing that I find quite frustrating is, first of all, we think our method is much better than others that are related methods, and people could look at the papers and look at the raw data and make their own mind up. But I really think we're having a slow start, whereas in the 3D genome area, everyone was like, wow, this is awesome, let's do all of it. I think this is now the time to do it with all kinds of RNAs. We should really get cracking it. It's really easy to get a lot of data, certainly with our methods and the other methods out there. Yeah, there's so many exciting questions to ask. Exactly. So, and I know Omar is very open to collaborations and also just to help other people just to get it going and try it out. It's really very simple. It just requires 2 cross-linkers, and that's more or less it, and a little bit of sequencing. But
So actually, as a follow-up to that, Jane Withka has a question. She wonders if your structures determined with comrades map well with those that are predicted by methods such as scan-fold and other RNA folding programs.
Yes. Yeah, so absolutely. So actually, so also sort of other chemical probing, like DMS or things like this, we find a lot of overlap. It's very comforting. And, but also with secondary structure prediction, I guess, so I guess the best would be to use a combination, right, of orthogonal approaches really to get the best picture. We just find, in particular, as we're trying, so my personal idea is that, if you could just find, for example, looking at cancer cells, you can find, some kind of reasonably stable structures, which is not going to be accurate, but things that we find using this method might be some cool structured RNAs in 5' or 3' UTRs that might be really interesting new targets, right? And there are a number now of interesting small molecules that it can at least interact with specific pseudoknots or G quadruplexes, if they exist in RNA, just joking. But, there is, I think that seems quite exciting to me.
Very cool. So there's another question from Ebru Kamak, who asks whether comrades is useful to detect small RNA interactions, or is this cross-linking efficiency not enough for that? So can you detect small RNA interactions?
Small RNA to small RNA. So we haven't done, I would think you could do it, but we have, essentially so far we've pulled down always something that's kind of long because we were also interested in the structural aspect. But I think we should be able to. Actually, we should look through our data. I'm sure we could find some microRNA, that is still, double-stranded or something, biogenesis intermediates or something like this. I'm sure we would find that. Yeah, absolutely. To be fair, there's a huge bias to AU base pairs, right? So if it's just GC, it's not going to work. Again, all of these methods have certain biases, but I think for most RNAs should be okay.
All right. A question from Sudeshna Fish. She asks, or he asks, given that miR-21 is upregulated in many inflammatory and injury-inducing conditions, do you predict miR-21 could be used as a biomarker of this viral disease? And then I would ask a second question. So, it seems like the virus might be sort of usurping this natural upregulation of miR-21 to its own advantage. So it could be biomarker as well as a potential therapeutic target.
Yeah, I completely agree. And we really would like to have a better model for this infection. So we use the human placental cell line, and we also use HeLa cells and a couple of other things. It's all very unsatisfactory. There are mouse models, and we started talking to a lab in Chicago to work on this together. But we haven't actually done it. I think that seems to me, most exciting to look in like in an animal model. So, and then essentially we could use a miR21 knockout mouse, or we could play this even to conditionally knock it out in different tissues. I think that would be more exciting than doing it purely in cell culture, because I'm just wondering that we are missing a lot, especially, for example, the immune response. Yeah, so yeah,
right.
Potentially exciting, but yeah, we haven't been able to get any data on it. Yeah.
Very cool. Are there any other questions for Eric? All right. Well, Eric, thank you very, very much for spending the time with us. I know the time zone has been challenging, but we really appreciate it. It's been great having you with us. Everybody else will be back next week. If anybody wants to sort of have a chat with Eric quickly, we'll keep the line open. Feel free to sort of hang on for a minute or two longer. Otherwise, we'll see everyone soon, hopefully. Bye-bye.
Well, thank you very much for the opportunity. It's very lonely right now, so it's always great to connect, even if it's just through Zoom. Thank you very much.
It's good to see you, Eric.
So Eric, if you have a moment, I would ask you another question.
Sure.
So, you know, there is a method called RNA antisense. You can use an RNA antisense as a probe also to pull down the interacting RNAs as well. I believe it has been published and used by Gutman, what's his name?
Yeah.
I'm sure you're aware. So I'm just wondering if you sort of had a chance to compare them to see if you, how would you compare them? Do you know if there are any advantages or disadvantages for each one of them? They're certainly both supposed to serve the same goal of identification of RNA to RNA interactions.
Yeah, I mean, maybe I shouldn't be too boastful, but I think, I mean, the method I think that we have is really at least an order of magnitude more sensitive than anything else that has been at least been published. But, you know, I think you would have to try it yourself. I mean, I wouldn't believe that me, but I mean, I don't know. But I think it's super efficient. It works really well. That the only problem is that I think it's not going to be very good at more, you know, RNA vicinity. It really needs to be proper base pairing. You need to have at least a little bit of a double helix, I think, to get a signal. And if you have an AU in there, then I think you have a good chance. Single AU should be enough. But and I hope I didn't insult anyone personally when I made fun of Sorlin. I don't really know anything about chemistry, but yeah.
Sorry, can I make a question?
Oh yeah, go ahead.
Okay, just a short question, Eric. Do you think it would be possible to design an aptamer to as a decoy to block miR21 action on Zika virus and block replication of Zika virus, and eventually for coronavirus the same thing?
Yeah, so absolutely. The only problem is, and Frank will know this much better than me, to the delivery, right? I guess lung might be not the worst place to start, but yeah, absolutely. So for the coronavirus, in fact, if you could mess with the FSE, I think, I mean, already by mutation has been shown, if you can mess with the frame shifting element, this is extremely detrimental to the coronavirus. And as I tried to say a few times already, I strongly believe that structured RNA is quite an exciting opportunity for potential small molecule therapeutics. But again, I'm not a chemist, so I'm just maybe a bit overexcited. But there are certainly antisense technologies. I think there would be lots of opportunities. I could see this anti-miR21 definitely. And in fact, we would really love to try this in a mouse, and yeah, and also for SARS-CoV-2. And I'm aware, in fact, that there are even a number of startup companies now that want to try, you know, antisense approaches for SARS-CoV-2. So yeah, I'm more a fan of small molecules rather than aptamers. But
well, Eric, if you don't get any takers on the miR21 knockout mouse, we have that in the lab, and like I said, we have.
Oh, really? Oh, excellent.
So if you wanted to, you know, sort of ping me if you don't get any takers, we can talk about it.
So was I correct in saying this? Was this just loose talk? But it's really a bit weird, the target space. Do you know more about it? It's
yeah.
mid-51. It's
Yeah, I mean, it is an enigma because it seems to be so important and upregulated under almost all perturbations, and yet the mouse is almost fine.
yeah.
So, but it hasn't stopped us going after it. I mean, we're still trying to target MIR21 with a variety
Okay,
of
cool.
approaches.
Ooh, excellent. Yeah, that sounds really exciting. Yeah.
So is your Zika paper already published?
Well, the Zika, yeah, I know. Everybody, I know. So to be honest, we tried to publish this, let's say, in a fancy journal, and we couldn't. So it was published in 2018 in Nature Methods. But because it's a method, I guess people don't get to the biology. Yeah.
Okay, well, we'll look it up. We'll look it up.
Yeah.
Fantastic. All right, any other questions for Eric? Okay, well, thanks everyone. It was good seeing everybody. And
yeah. And feel free to be in touch using, other means like email and stuff like this.
Sure, sounds good. I'm going to send you the email about the RNA Collaborative.
Brilliant. I'm really looking forward to this. Great.
All right. Bye, everybody.
Thank you very much. Bye.